Hello, my name is Ellen Streitwieser, and this is my final project presentation for Chem 394. Proposed steps for investigating method of action of sertraline ligand binding to SERT protein. The area and problem that I chose to focus on for my project is the method of action for SSRI ligands binding to SERT protein. SSRIs are selective serotonin reuptake inhibitors. They are a class of medication known as antidepressants, and the SSRIs specifically act to inhibit serotonin transport proteins called SERT proteins. SERT proteins are responsible for transporting serotonin from the synaptic cleft back to the presynaptic neurons. They inhibit the reuptake of serotonin, which achieves a therapeutic effect of increasing overall serotonin levels. The interesting thing about SSRIs is that their specific method of action is still not known. The binding site for SSRIs is an area of active research as far as trying to investigate the actual amino acid residues that are involved in creating the binding pocket for the ligands, the SSRIs, to bind to the protein, which is the serotonin transporter protein. For this project, I'm focusing specifically on sertraline. Given the time frame and my knowledge base, I chose to narrow it down to just one SSRI in particular. Current research into the binding sites for SSRIs looks at the molecular structure of SSRIs for specific atoms that would contribute to binding. So there's two areas that have been investigated. One is looking at the halide substituents on the SSRI ligands. Now, the different halides on the main molecule is what differentiates one SSRI from another. Sertraline is the molecule that has two chlorine atoms, and that is the one that I was looking at. The second area of study, which is where I focused on in my project, as well as the resources that I found, is looking at the nitrogen that is part of the SSRIs and the surrounding amino acid residues, and looking at proposed salt bridges that form between these amino acids and the ligand is a way that stabilizes the binding conformation of the ligand to the protein. And it does this by the ionic interaction between the amino acid residues and the nitrogen on the ligand, which is lowers the energy of both molecules. So the first resource I used was antidepressant specificity of serotonin transporter suggested by 3 LuT SSRI structures. In this study, they took a bacterial homologue of the SERT protein, which was the leucine transporter sequence, LuT, from Aquifex aeolicus. I hope I'm saying that right. And they did this in order to look at proposed binding sites for SSRI medications, sertraline, R-fluoxetine, and S-fluoxetine. As LuT is a bacterial homologue of the SERT protein, it has very similar, if not identical, amino acid residues, which makes it an excellent model for studying the binding sites for these SSRIs. So the sertraline in this study was targeted based on the salt bridges that is proposed to form with the nitrogen on the SRI ligand with 2 aspartate amino acid residues. And for the bacterial homologue, it's aspartate 401 and 404. So looking at a salt bridge that forms between the ligand, these 2 aspartate amino acids, and the water molecule. Now, interestingly, for this study, they didn't alter the aspartate amino acids to chest function, but rather they altered an adjacent residue to the salt bridge, which was GLU 493. They dubbed this a gate residue, as it is key to the function of the salt bridge. So this amino acid residue was mutated to an aspartate, and it was found that the total binding affinity decreased by a factor of 15. Now, the significance of a study like this is if you can alter the amino acid residues used in a protein and show a change in the overall binding affinity or energy or conformation of the protein, then you have shown that the amino acids specific to the wild type do play a role in the binding of the ligand initially. So this is a figure from the article, and they investigated, again, 3 SSRIs, but the sertraline is the molecule in yellow. And so what they have marked as figure D shows the proposed salt bridge between the nitrogen of the sertraline and the aspartate 401, and aspartate 404. The second resource that I used was identification of the inhibitory mechanism of FDA-approved selective serotonin reuptake inhibitors, insight from a molecular dynamics simulation study. So this paper was very interesting because they also use molecular dynamics software. They, instead of looking at a bacterial homolog, they took the actual SERT protein, a computational model of it, and mutated specific amino acid residues to check for a difference in binding affinity. So they looked at TRY95, ASP98, isoleucine 172, ASN 177, PHE 341, and serine 438. They tested mutating these various amino acids and along with a variety of SSRIs. Looking specifically at what they did with sertraline, they were again targeting the proposed salt bridge, but they were saying, they guessed that the salt bridge is formed between the ligand and serine 438 and aspartate 98. So this is different than the last study, which proposed that it was between two aspartate amino acids. And in this study, they mutated the serine to, they mutated the SER 438 to a THR residue to test for binding affinity. Now, the single mutation showed a decrease in the electrostatic and van der Waals energy, as well as a decrease in free energy of the surrounding solvent. This is a figure from the paper, and image B is sertraline. And again, they have shown here the proposed salt bridge formed between the aspartate and the serine amino acids, as well as the nitrogen in the sertraline. This is a table of their results showing the difference in energy levels in Gibbs free energy between the wild type and the mutated amino acid residue. So the commonality between these two studies is that they both agreed that the aspartate 98 residue was involved in the salt bridge. And so that is where I chose to focus my project. So the general plan for my proposed solution is to load a computational model of biologically identical SERT protein with the SSRI ligand of sertraline, noted as SRE, to mutate a suspected binding site for the SSRI, sertraline, measure the thermodynamic alteration between the wild type and the mutated molecules, evaluate the results to determine possible energetic changes in the mutated protein and ligand binding site, and if mutated residue triggers a statistically significant change in energy output and or the protein conformation, it could indicate a possible correlation between the target residue and the ligand protein binding site. And the purpose of this is to try to determine the method of action of SSRI binding to SERT protein. So this is an overview of the demonstrations of steps that I did for this project, and then I'll go into them in more detail. I found a PDB of the SERT protein and the ligand. I converted that from a protein database file to a protein structure file and mutated residue 98 from aspartate to valine, with the goal of disrupting the salt bridge that stabilizes the conformation. To do this, I had to find the proper charm file, as well as generate one for the ligand in order to have the proper topology and parameters information. I uploaded this in order to convert it in VMD and use QwikMD as well. Then I mutated the residue 98 from aspartate to valine, loaded these molecules, both the wild type and the mutated, into Avogadro and generated the Gaussian input files. The next step was to run a geometry optimization, single point energy, and frequency output through Gaussian 16. However, I ran into an error due to a lack of memory, and given the time frame of this project, as well as my own knowledge base, this is where I stopped. Okay, so first I downloaded the protein database file for SERT protein that included the SRE ligand. And I found this on the RCSB protein database, and the ID was 6AW0. And it was the TS3 human serotonin transporter with sertraline at the center site. These are images that I generated in VMD after loading in the PDB of this file. On the left, you see the overall protein structure, and it's coded based on the different sections. And on the right, you see the alpha helices of the chain A of this protein, and in the middle of it, which is the proposed binding site, is the sertraline ligand. So the SRI ligand I built within Avogadro. I did a geometry optimization to make sure that my bond angles were correct, and I was able to export that as a .mol2 file, which I uploaded to the University of Maryland's CGenFF in order to generate the .str for the SRI ligand. This allowed me to upload the topology and parameter files within QwikMD, which was necessary in order to generate the protein structure file. So you can see this molecule is mostly just carbons and hydrogens. However, it also has a nitrogen, which is marked in blue, which is the target area for my project, looking at the ionic interactions between the nitrogen and oxygen in aspartate residues, as well as in green is the chlorine atoms that make this SSRI sertraline. So I converted the PDB to the PSF using the VMD extension automatic PSF builder once I had loaded in the topology and parameters files, and I mutated residue 98 from aspartate to valine. And the reason that I chose valine as my mutated residue is because aspartate has oxygen atoms which have an ionic interaction with the positively charged nitrogen in the sertraline. So by taking the amino acid that had the oxygens available to have this interaction and changing it to a non-polar substituent that has just methyl groups off of it, it would disrupt the proposed salt bridge that would stabilize the conformation and the binding of the SRI ligand to the SERT protein. So on the left here is the wild type shown in VMD. The overall protein is in blue. However, I have highlighted the aspartate residue specifically, as well as the ligand in the center. And on the right is this mutated molecule that shows the valine in its place. I measured the distance between the atoms in VMD. This is prior to optimization, so the only differences in length here are just shown between the specific amino acid residues themselves. This was meant to be a baseline measurement to compare to after the structure was optimized. As well, I took measurements of the surrounding amino acids just to have again as a baseline. On the left is the wild type, on the right is the mutated. And the distances are the same between the ligand and the surrounding residues. My next step was to run these molecules through Avogadro and generate the Gaussian input files. And I attempted to run the input files through Gaussian 16 to get a geometry optimization, a frequency, and a single point energy. However, I had an error due to lack of memory. I am not sure whether this is lack of memory on my computer that I was running these jobs through, or if it has to do with the way that I ran it through the ASU compute banks, or perhaps what was allotted to this type of job. I'm not sure, but this is as far as I got. With more time and more knowledge, I think I would have been able to do more with it. You definitely could run these to get the output files, and the significance here is that looking at the output after optimizing it would allow you to see, for example, if I go back, if there was a difference between the ligand and the surrounding amino acids. Because if you're able to optimize the structure and show that you had an overall conformation change of the protein, that would alter the binding site. And particularly if you also have a change in the energy of the system between the ligand and the protein now that it's been mutated. Because the reason that the aspartate amino acid was targeted as a potential contributor to the binding site is because a salt bridge between this amino acid and the nitrogen would lower the energy of this binding, which would increase the binding affinity. That is where I was headed with this project. And so as a result of how far I got, my proposed next steps were to optimize both structures, both the wild type SERT protein with the ligand, as well as the mutated protein with the same ligand, run a Gaussian frequency, geometry optimization, and single point energy output at the B3LYP 631GD level of theory. Compare the 0 point energy of the optimized structures as well as the Gibbs free energy to see if mutating the aspartate 98 to valine changed the energy states of the interactions between the SERT protein and the SRI ligand. and then measure bond angle, length, torsion of the amine group of the SRE ligand and the residue, both the wild type and the mutated, to see if mutating it and then optimizing the structure disrupted the overall conformation of binding between the ligand and the protein. These are the references I use for my project, and that concludes my presentation. Thank you.